Right, we're going to wrap up our view of brain imaging technologies with a few random observations. First off, we want to just remind ourselves of the contrast between fMRI, which is possibly the best known of these neuroimaging technologies, and EEG, which is the oldest of them. In fMRI, the signal that we record doesn't register brain activity directly. It registers regional blood flow. It measures the oxygen content of blood flowing through the brain. So it's a very indirect measure of neural activity. Because we're using all the powers of an MRI scanner, we have the ability to localize the signal very well. We can say where something happens down to less than a cubic millimeter usually. But one of the prices we pay for that is we have very poor resolution in time. We can acquire a scan over about 15 seconds, and we can't make any finer distinctions than that. In 15 seconds, an awful lot happens. Contrast that with EEG, the oldest of neuroimaging technologies, where we're measuring electrical activity at the scalp. A dreadfully messy signal, very difficult to interpret. But one thing it has is very good time resolution. When something happens, we register it immediately. So we can say when something happens in time to less than a millisecond. Compare that with 15 seconds for fMRI. But on the other hand, we have a dreadful time saying where it happened. We might be able to identify the quarter of the brain in which it happened, but that's about as good as our spatial resolution gets. So there's quite a trade-off here. And so you can see different technologies bring different advantages and disadvantages with them. Here's a fairly recent technology which seems to combine sort of the worst of both worlds. This is near-infrared spectroscopy, or NIRS. Here, it's also a non-invasive technique. It also uses a swim cap, much like EEG, but instead of registering electrical activity, what we have are pairs of paired elements, one of which is a light emitter, an LED emitting near-infrared light, and the other is a sensor recording the light emitted by the first one after it's passed through the skin, the skull, the top layers of connective tissue, and a little part of the brain. The light signal that's recorded then will vary as the blood flow in that top level of the brain varies. So like fMRI, we're not recording neural activity directly, we're recording regional blood flow, so it's a very indirect index. Like EEG, we're recording from an absolutely random subset of the brain, that part of the cortex that happens to lie near the surface. So it's a terrible, terrible signal. It's a relatively new technology, and the reason that we work on technologies like this is that we see advantages to combining them. So you can build EEG sensors and NIRS sensors into the same swim cap, and you can record multiple signals of multiple kinds from the same individual at the same time. And a lot of work now is examining the possibility of combining neuroimaging technologies together as we study people carrying out various kinds of tasks. But there's one other kind of combination that we can do, and this is the more interesting one. We can combine people. We have begun to find ways of recording from one brain, from multiple brains at the same time. This is known as hyperscanning, and it sounds very futuristic. In an EEG context, this is fairly straightforward. We can just take the signal from multiple head caps and record them simultaneously. And then with EEG, you can do some behavioral tasks. You can interact with computer screens. Talking interferes with the signal, but that can be kind of overcome. You're not going to be out there playing football wearing EEG, but you can do some tasks together. NIRS has it similarly allows you to engage in certain kinds of activity. fMRI is going to be much more difficult because individuals are inside scanners and they're not allowed to move. What's of interest here is that we've recently realized that brains behave differently when we interact with each other socially. This is very, very important. Our fundamental understanding of what brains are and what they do is constantly changing. And our recognition that brain activity needs to be sensitively interpreted together with the context in which it occurs, especially when that context is other people interacting with other people, this is a very important insight. So important that some have dubbed it the dark matter of neuroscience. There's all this stuff that we have not noticed up to now, and this is very recent work. So in social interaction, when we do stuff together, our brains literally become coupled. Your brain and my brain become non-independent. Now there's lots and lots of challenges here. With fMRI, we've got a very slow signal. As I said, it takes about 15 seconds to do one scan. So we're not going to be studying any kind of coordination of movement. And also subjects can't really move in the scanner. They can push a button to signal yes or no, and that's about all they can do. So it's suitable only for studying abstract forms of coordination. But there is a lab, at least one lab, with multiple MRI scanners in the lab. And you can put people in and have them play abstract games like Prisoner's Dilemma games. These are kind of games studied by psychologists who want to understand processes of social affiliation and decision making. Imagine, if you will, that there are two prisoners and they're locked up and they're going to be interviewed separately, and the authorities want something from them, and each prisoner must decide for themselves whether they are going to cooperate with each other and stay silent, or whether they're going to defect and rat on their colleague. And there's different payoffs for different behaviors. Remember, they're separated from each other. If they both stay silent, they both serve 1 year in prison, let's say. If prisoner A rats out prisoner B, and prisoner B stays silent, prisoner A walks free, and prisoner B gets 3 years in jail, much worse. But the same holds the other way around. If A stays silent and B rats A out, then B walks free and A gets 3 years. And if they both rat each other out, that's a terrible outcome. They both serve 2 years. So obviously, well, it looks like the best thing for them to do is to cooperate with each other, that is, both stay silent. But if they both know that, then they each know that, hey, I could go free if he's going to work on that basis. So these are interesting games that people play, and you can do that kind of decision making in a scanner. But fMRI is only going to go so far. EEG and NLRS are much more promising here. They give much better temporal resolution, and subjects are free to move. This is the important thing. The spatial resolution is lousy, and signals are very weak. But an awful lot of work is going on in looking at ways of combining these methodologies. So finally, I'd like to look at one other use that we can put signals that we record from the brain to, and that is the wacky world of brain-computer interfaces. Now, don't get too excited. People tend to understand far more from the term brain-computer interface than is warranted. Why would we hook a brain up to a computer? Well, actually, let's start off by being very, very clear. We already have a brain-computer interface. This is it. It's my fingers. And they work on the keyboard, and that's how I interact with the computer. That's a brain-computer interface. The brain itself, that organ in the head, was not really... it's going to be very difficult to get that to interact with a computer. We can put electrodes on, but I can't consciously affect those electrodes. Well, I can do a little bit. We'll see what we can do. But there are reasons you might want to do this. It is primarily to facilitate communication for people who have severe difficulty with moving. People with neurodegenerative diseases, locked-in syndrome, ALS, and similar neural diseases that stop them moving. If you can't move any muscles, you can still be conscious, and this is a means of restoring some kind of communication. That's the principal motivation here. Similarly, signals read from the brain and from the peripheral nerves can be integrated into technologies to help restore mobility in cases of paralysis and in cases of neuroprosthetics. So if we give you an artificial arm, trying to find ways to use the signal from the nerves to move that arm, those are very, very important uses as well, slightly different focus. A lot of people think of gaming uses, trying to control console games using an EEG, and there is a small market for that. It's nonsense. Don't worry about that. And there's also, of course, a lot of this work has, as usual, been funded by the military. I'm sure the military have their own purposes here, but most of the work here is concerned with communication and neuroprosthetics. So if we can record a signal from the brain or from the nervous system, we can use that for in various ways. The signal is going to be really messy. We're going to have to record it and then try and analyze it and use that for doing various things, like, for example, controlling a wheelchair or moving a mouse on a screen. So the brain signal conveys somehow intent. We're going to understand the brain signal as conveying intent. If we're recording directly from the cortex, we're bypassing all the peripheral nerves and muscles, and progress is very limited so far. Here's an example of what it looks like to write a message using a brain-computer interface. Imagine you're looking at the screen and the dot comes in from the left and the dot starts moving from left to right. And you have learned to influence the trajectory of this dot by thinking in one way or another. And you can steer the dot towards one of 4 blocks of letters there on the right-hand side. So you can choose the top block, which is the letters A to I. At that point, the screen changes and the dot reappears on the left and comes in and moves across the screen. And now you're selecting among the letters ABC versus DEF versus GHI. Again, you select one of those boxes and now the dot reappears on the left, comes in and moves slowly, and now you're going to steer that dot with your thoughts towards one letter. Congratulations, you just typed a single letter. And now we're going to do it again to type the next letter. This is not a fast means of communication. This is dreadfully inefficient. You use it when it's the only thing that's available to you. You can learn to control this signal to make what is in effect a 1-bit discrimination. You can move that signal towards the top or towards the bottom. Shown there is the EEG signal, in which there's a dominance of one side of the brain rather than the other. And shown on the right-hand side is the signal that differs depending on whether you're aiming at a top target or a bottom target. But you can see this is not going to be an efficient way of communicating. It's far less efficient than your brain-computer interface here, which is probably millions of times faster. This is an infant science. Non-invasive techniques are preferred, but remember, sometimes here we're dealing with people who are very, very severely impacted by a medical condition. And it might be actually worthwhile, in such cases, opening up the skull and placing electrodes directly on the brain in order to improve the signal. And the signal you can record directly from the cortex is going to be much better than the EEG signal that we mentioned, for example. So when it comes to this kind of technology, brain-computer interface, we got to restrain our imaginations. Science fiction movies present this as being an entirely different class of problem. Imagination runs much faster than technology, and the technological development here is very slow. So that wraps up what we're going to say about brains. Some things to think about after this are that it's not easy to find the right questions to ask of brains. We're not only trying to find out what the brain does and how it does it, we're trying to find out what's the question we should be asking in the first place. And in what context should we be asking that? We are at the very, very beginnings of our inquiry here. It's 2020. Maybe human species will be wiped out before we make much progress. I don't know. But we've a long way to go. When we are so ignorant, then convergent evidence from multiple paradigms is much more satisfying than anything you can get from a single study. So we can learn something from an EEG study. We can learn something from an fMRI study. We can learn something from a behavioral study. We can learn something from theory. We can learn something from modeling. Our knowledge becomes more secure as we triangulate using all of those. At the start of this, we noted the mistake of the pseudoscience of phrenology in attributing specific functions to specific parts of the brain, saying this is the part of the brain for X. We are constantly in danger of repeating that mistake, especially if we focus on results from a single neuroimaging technology. We ought not to repeat that mistake. Right, so that concludes our survey of brains.